Hi, my name is Keith Smith. I'm the first author on the paper Hierarchical Complexity of the Adult Human Structural Connectome. And I just want to give a brief overview of the highlights of the paper. So this is based on some novel network science methods. So hopefully you understand that a network is composed of nodes connected together by edges. And for example, another basic property is that each node has a number of edges which it connects to other nodes in the network, and this is called its node degree. So in network science, we have this basic problem. We have many connections, but it's very difficult to compare, to understand what's the difference between different networks that we find just by looking at them. They look quite messy. So this is where topological tools come into force. So here are some examples of very ordered networks. There's a star network, a regular lattice, grid lattice, and a fractal modular network. And in the beginning, I wanted to understand what is it that unifies the sense of orderedness in these networks. Is there anything we can find which can very efficiently explain that each of these networks has a high degree of order? So if you'll notice, and what I noticed was that basically nodes with the same degree have highly homogeneous neighborhood degree sequences. So the degrees of the nodes that these nodes connect to are basically the same in these examples for nodes of the same degree. So in the top left, the star network, all nodes of degree 1 have a neighborhood degree sequence of 8 because they all connect to a node of degree 8, and they all just have degree 1. In the grid lattice, we have corner nodes of degree 2, and they connect to 2 nodes of degree 3, so 3, 3. So we see a very homogeneous neighborhood degree sequences for nodes of the same degree. And this is what I thought, okay, we can characterize this by looking at the variance of these sequences. So if we look at if we lay the sequences out, and then for the sequences of the same degree, they have the same length. So then we can basically take the variance vertically along these sequences. So all the variance of all of the first elements of each sequence, and then the variance of all of the second elements of each sequence. And we take an average over the number of elements in the sequences. So in each of these networks, we get this value of 0. But we want to understand this in the brain. Is the brain similar, or is it much more heterogeneous? So for a network, we propose this measure of hierarchical complexity. And in general, we can think of hierarchical complexity as the variability of roles played by agents in an equivalent hierarchical position for a given system. So a very familiar system which we all are familiar with is the hierarchy in academia. So we have lecturers, postdocs, PhDs as a very simplified model. So they may all have an equivalent, nearly equivalent salary, but the reality is, for a successful organization, all of these people are not doing the same thing. Instead, what we expect, or what we hope to see, is that at each level there's a variability of the talents and skills that each person is bringing to the team, which allows for a complex organization. So here's another example of a hierarchically ordered network, and then we can basically arrange the edges so that this is a much more complex, hierarchically complex network on the right. So again, can we spot the difference between these networks? The first network is a random network. The middle network is derived from a structural connectome. And the one on the right is basically a structural connectome where the edges are randomized. So the degrees of the connectome are fixed, but the edges are randomized around the network. So again, how do we distinguish these kinds of networks? So we follow a pipeline where we take the MRI signal. There's MRI signal processing done by my collaborators. This gives us some binary adjacency matrices, which we can basically generate random null models from. We took 3 random null models in this instance. We took the Erdős-Rényi random graph, which is a highly integrated network, and integration is always touted as a very important feature of brain networks. We also looked at the random geometric graph because also we see in brain networks a tendency for connections to make much more local connections than across across the brain. Although people also say that these long distance connections are very important for integrating the information in the brain, but in general, there's more local connections than these kind of global connections. And the third binary random null model we look at is this edge randomization approach, which is called the configuration model, which we saw in the previous slide. We then analyze these networks for globally. We look at global properties of the network, but then we also wanted to look into, see if we could prize apart some hierarchical tiers, notable hierarchical tiers in the brain networks, and see what the complexity analysis that we have developed can say about this and how it's important in the brain. So we applied these to our. We've applied a range of different network indices to a range of different models. So on the top left is a hierarchical complexity, and we see very clearly that the human MRI is more complex here than all of the null models that we've generated. And there's a kind of spectrum almost from going from the random graph, the completely random graph, to the random geometric graph, where we've got geometric localities fixed but randomized in the geometric space. And then we've got the configuration model, which is slightly more complex, but the most complex is the human MRI. On the top right, we've got degree variance. So this is a measure of the heterogeneity of degrees in the network. So this doesn't prize apart the human MRI from the configuration models because the degrees are fixed in the configuration model. We also have assortativity, which is kind of a correlation of degrees which are connected. So are degrees more likely to be connected? Are nodes more likely to be connected to other nodes of a similar degree or not? We see that in the brain, in our analysis, there wasn't any strong sense of assortativity or disassortativity, and was fairly similar to random graph models. On the bottom right, we looked at the integration segregation using the normalized clustering coefficient. And again, we see that actually the human MRI and the random geometric graph have very similar levels of segregation, actually. We then also wanted to see what are indices correlated with other indices, with these other indices and even other indices. So we looked also at the Rich Club coefficient and the characteristic path length, that's K and L, and also C, R, and V. There's a clustering coefficient, assortativity, and degree variance. And it was quite striking actually that our hierarchical complexity metric was fairly uncorrelated compared to all the other networks that we studied, which kind of points towards that maybe these other network metrics are just telling us about a single topological property, actually, which was surprising. So again, here's another example trying to kind of illustrate what is hierarchical complexity. So we have some random null models, and we also have a structural connectome on the bottom left. And we're looking at a single degree, nodes of a single degree in the network. So in this case, we choose degrees of 31, I think, just because all of the null models had degrees of 31. So it's quite clear from this picture, I think, what we're getting at. These random null models, the nodes of the same degree actually have very similar neighborhood degree sequence structure. It's much smoother. You can see that these images are much smoother for these than it is for the structural connectome, where we get a much more variable, diverse range of degree sequences. So then we broke our networks down into 4 tiers. And this was just based on the maximum degree. So we took the nodes which had the top 25% of the maximum degree, and then in tiers with 25% basically. So it was quite interesting that when my collaborators looked at this, he was able to see basically that the tiers corresponded broadly to known functional processes, different functional processes. So in the top tiers was related to cognition, which is fairly well known. But then we also see a second tier, which is related to sensory motor processing. Below that, there's a third tier, which is cognitive sensory integration. And then below that, possibly related to memory and emotion. And this is based on a paper by Sepulchre et al., the multimodal organization of the human brain. So we looked at the complexity within these tiers. So all degrees within each tier, we looked at the complexity within the connectomes for each of our 79 individuals. And we also did this we did it for a 4 tier and also an 8 tier, but the 8 tier was not very consistent, whereas the 4 tier seemed to have much greater consistency. So it seemed like a more reasonable method, more reasonable paradigm to use. The result basically indicated that the top tier was not statistically, the means of the distributions between the connectome and their configuration models in the top tier were not statistically different, although there was obviously higher variance in the human connectome than in the configuration model. But it was interesting that the complexity was on average about the same. Similarly was the case for the 4th tier, the lowest of the lowest degrees. Not we couldn't find a statistical difference in the means, but tier 2 and tier 3 both showed quite strong statistical differences in their means. So this is quite interesting. So if we relate this back to what we have before, so we see that the cognitive processing, memory and emotion, the nodes involved here, they don't vary so much in the kinds of connectivity patterns they make. Whereas in sensory motor processing and cognitive sensory integration, we see that there's much more variability in the connectivity patterns made by the nodes in these tiers. And here is, we've mapped these tiers to the brain. So we've got the cognitive related tier and is the blue, the sensory related tier in the green, the cognitive sensory integration is the orange, and the kind of memory and emotion is in the red range. So this is what it looks like on the map, basically, where the red implies relative to our configuration models. The red implies that the node connects across the hierarchy. There's a much greater variance than the configuration model, and the small variance implies that the node has a much more limited range of the connections that it establishes in the brain network. So we looked at some notable nodes, notable ROIs, regions of interest in the brain, which seem to have very strong connectivity patterns, strongly different from what would be expected. And it was quite interesting. We see the left rostral anterior cortex was attached strongly to the cognitive tier 1 and the memory and emotion tier 4. Then this agrees with its known unique role in cognition and emotion. The entorhinal cortex was the only region with overrepresentations of tier 3 nodes, which is cognitive sensory integration, which lines up with the knowledge that superficial layers receive both multimodal and unimodal sensory inputs and project to widespread cortical and subcortical loci. And then we also looked at superior parietal gyrus, for example, the large representation of tier 2 nodes, sensory motor processing, aligns with its known predominant function in these processes. So our conclusion is that the topological hierarchy of brain structure appears to be broadly linked to functional processes. The human brain structure is very hierarchically complex compared to our null models. And also we can see this as a distinguishing feature of the brain topology as opposed to other network indices. The complexity of the brain is explained by a variety of different roles played by these different regions connecting in different ways throughout the hierarchy. And the variety of connectivity patterns across the hierarchy shows alignment with known functional roles of the ROIs. Okay, thank you very much. We wanted to then characterize what are these different roles and how can we relate it to this tier structure that we found. So what we did was basically we could take a measure of the variance of the sequence for an individual node across all of our participants, all 79 participants. So a greater variance here would imply the node connects across the hierarchy, the degree hierarchy, and a small variance implies the node is connected in a much more limited